Hey A&Pers, this is Dr. Jones checking in. It is time for an A&P1 lab video. So this is lab video 1. We're going to start on page 24 of your note set. What are we calling this? Laboratory guide. There we go. So we're going to start on page 24. These material are seen on the slides and also on these models. So this is the skeletal muscle model. Each cell, each pool noodle is a single skeletal muscle, a single skeletal muscle fiber. And you see these are all nuclei and the little lumps just under the sarcolemma. Sarcolemma is the cell membrane of a skeletal muscle cell. Striations will be these red stripes. Endomysium is this rough connective tissue that separates and electrically isolates the cells. So all this mortar between the cells is your endomysium. Now, it's not on the note set, so it's not a correct answer on the lab test, but people often ask, and these blue blobs in the endomysium are going to be your fibroblasts. Remember, connective tissue is the cells and the extracellular stuff they make, which is largely fibers, and they're made by these cells, the fibroblasts. All the red tubes will of course be your blood vessels, and here you have an axon and a neuromuscular junction. Next is cardiac. Each cardiac muscle cell is a cardiocyte. They are striated. They have a single central nucleus, or at least no more than 5. They have endomysium, fibroblasts, still not on the list, sarcolemma, blood vessels, axons. But the important thing here are the intercalated discs. The intercalated discs are special because they have holes in them called gap junctions. The gap junctions allow ions and other small things to move directly from cell to cell. The intercalated discs also connect the sarcomere from one group, the myofibrils, to this next cell, so that this heart can beat in a coordinated fashion. Because the function of the heart is to pump blood, if the muscles are just all contracting but not in a coordinated fashion, you won't pump enough blood to feed the whole body. So the heart, because arteries come out the top of the heart has to beat from the bottom up, and how it coordinates that are these black lines, the intercalated discs that connect the cells. The cells are called cardiocytes. Connective tissue covering, endomysium, and the gray ovals, nuclei. And that leaves smooth muscle. Smooth muscle is spindle-shaped or toothpick-shaped. They have a single central nucleus. They are not striated. They do have actin and myosin, but it's shaped kind of like a net, not sarcomeres. So still endomysium, still blood vessels, fibroblasts, axon. So all the same stuff. That was page 24. I'm going to flip to page 25. Zooming out. Okay. So this is a single sarcomere. One sarcomere. Did I just say sarcomere? One single skeletal muscle fiber. Yeah, y'all caught it. So this is a skeletal muscle fiber. We're at the top of page 25. The first structure is the rough connective tissue, which is the endomysium. Next is the smooth cell membrane, sarcolemma. And then each of these and these and these are called myofibrils. If you need to know, each group of myofibrils is called a Conheim's area. And then the sarcomere is from Z line to Z line, Z line to Z line, Z line to Z line. Sarcomere, 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 sarcomere. We'll cover why that is in a minute. The nuclei, there's a nucleus here, nucleus here. You can see that from the side. You can see this one from the side. And this is the axon. Now, on the off chance of the test, they tag this part. We're going to call it the axon, but we know that the axon is the center structure with the number 8, and the tortillas wrapped around it are a different cell that we will learn for lab test 4. So the axon is part of the neuron, and the neuron ends at these gecko toes, and these gecko toes are called, yep, you've got 3 choices here: synaptic knob, synaptic terminal, or axon terminal. So any of those 3 are correct. Synaptic vesicles aren't on this model, and the synaptic cleft is this gap between the nerve cell and the muscle cell, this gap right here, between the nerve cell and the muscle cell. And then the motor end plate is this pink wiggle squiggle. And you can also see the motor end plate here, where the synaptic knob has been removed, this pink wiggle squiggle. There you go. And that's a specialized area of sarcolemma, and it's specialized in that it's A, folded, and B, has a boatload of neurotransmitter receptors. What neurotransmitter is released from the synaptic knob? Acetylcholine, which is abbreviated capital A, capital C, lowercase h. So that's everything on this model. We're going to start over at the top of the same page with the newer model. So this is, again, one single skeletal muscle cell or skeletal muscle fiber. And we start with the rough connective tissue covering, which is the endomysium. The smooth cell membrane, which is the sarcolemma. And then each rod dissected out or cut is a myofibril. So the myofibrils are a different size. So here's a single myofibril between my thumbnails. And now here's a single myofibril between my thumbnails. So the cell is the same size, but the myofibrils are at a different scale. So we've effectively zoomed in, even though we kept the cell the same size. So the scale here on the older model is slightly more accurate, but with this zoom, we can see more detail. So we're gonna use both. Let's see, myofibril we did. Okay, sarcomere is from Z line to Z line. So here's a sarcomere, here's a sarcomere, from Z line to Z line. We'll come back to that in a second. There's only one nucleus on the cell. The axon is the yellow center part, and the tortilla surrounds it, is wrapped around it. Again, if they point to this other cell that surrounds it, for this test, we'll call it an axon, but for the next test, neuro, you need to know exactly what that is. And so the synaptic knob, synaptic terminal, axon terminal is this whole yellow splotch around. And here we see the synaptic vesicles. You see the pearls? Each pearl is a synaptic vesicle. And the synaptic vesicles contain neurotransmitters called acetylcholine. And when this nerve, the axon of this nerve, brings a nerve action potential, it causes these synaptic vesicles to undergo exocytosis, which is when the cell poops, and that releases its contents into this baby blue gap here, which is the synaptic cleft. The synaptic cleft is the gap between the nerve cell and the muscle cell. So yellow here is nerve cell, wiggle squiggle in is muscle cell. So when the acetylcholine is released into the synaptic cleft, half of it is broken down by the enzyme that breaks it down, acetylcholinesterase, which is abbreviated capital A, capital C, lowercase h, capital E. what gets across the synaptic cleft, the acetylcholine from the synaptic vesicles that gets across the synaptic cleft, binds to receptors, acetylcholine receptors on the motor end plate. And again, the motor end plate is a specialized area of sarcolemma in two reasons. A, it's folded, and B, it has acetylcholine receptors. So when the acetylcholine binds to the acetylcholine receptors, those receptors are chemically gated sodium ion channels. So what chemical opens them? The acetylcholine. Does the acetylcholine go into the cell? Heck no. The acetylcholine does not go into the cell. The acetylcholine just binds to the receptor and opens it. So what goes into the cell? Sodium. Lots and lots of sodium. And those positive ions rushing into the cell from outside the cell are your change in the transmembrane potential, which is the muscle action potential. So then that muscle action potential sweeps down the entire sarcolemma of the entire length of the cell, and this is just a small chunk of a cell, because remember, these are long and skinny, like hundreds of pool noodles stuck together, okay? And then that muscle action potential that is sweeping down the sarcolemma, cell membrane, sarcolemma, also goes inside these transverse tubules or T-tubules. So these T-tubules are the blue on this model, and they bring the excitement, the muscle action potential into the cell. And each of these T-tubules is touching two terminal cisternae. So the T-tubule with the two terminal cisternae, these three structures together, are called a triad. And the terminal cisternae is the cuffs of the lacy sweater sleeve. And the whole lacy sweater sleeve is called the sarcoplasmic reticulum. And the sarcoplasmic reticulum, along with the terminal cisternae, stores boatloads of calcium. I believe our book said there was 40,000 times more calcium in the sarcoplasmic reticulum than there is in the sarcoplasm. So here's the deal. The terminal cisternae touching the T-tubule, remember the T-tubule carries the muscle action potential, which is an electrical activity inside the cell. And these terminal cisternae have voltage-gated ion channels. So the muscle action potential, which is started by the axon, has a nerve action potential, which releases acetylcholine from the synaptic vesicles into the I'm off screen. Okay. Nerve action potential at the synaptic knob causes the exocytosis of acetylcholine from the synaptic vesicles into the synaptic cleft. They bind to acetylcholine receptors on the motor end plate, allowing sodium to rush into the cell. That muscle action potential sweeps down the entire sarcolemma and into the cell on each T-tubule. So the excitement at the T-tubules opens voltage-gated calcium ion channels. Remember, voltage-gated are activated by a change in voltage, which will be an electrical activity like, say, an action potential. And that causes the release of calcium into the sarcoplasm from the sarcoplasmic reticulum. All right, and we're at 14 minutes, so I will stop this video and we will start again with the ugly model. And this is A&P 1 Lab Video 1 for Lab Test 3. The education must go on. Dr. J out.